This is a presentation on the organism Acinetobacter soli. The presentation will cover characteristics about A. soli and how it is pathogenic and has the potential to cause outbreaks. Acinetobacter soli is a non-motile, rod-shaped bacteria belonging to one about 47 other Acinetobacter clinical isolates that are very problematic. Acinetobacter soli is a gram-negative, oxidase-positive, and catalase-positive organism in which catalase enzyme is present, releasing oxygen from hydrogen peroxide. And this helps to differentiate from non-catalase producers, such as Streptococci species. Catalase testing is very important to differentiate both A. soli from Streptococci because these isolates both produce bloodstream infections. However, Acinetobacter soli is our main focus here. As with many organisms, Acinetobacter soli has a place in where it is isolated. This organism is typically isolated from forest soil originally from the soils from the mountains of Baekgun, found in the Republic of Korea. This sounds reasonable because the organism was isolated from spinach leaves during the True Unknown section. During the past 24 years, Acinetobacter species have been reported to cause infections causing pneumonia, sepsis, wound infections, along with other opportunistic infections caused by these organisms. These infections are mainly nosocomial infections, which is basically found in hospital settings, which is a problem because it potentially can pose risk to patients, visitors of the hospital, and other healthcare workers. Treatment for most of these infections caused by these opportunistic pathogens are difficult to treat, as A. soli is highly resistant to many antimicrobial agents. During past years, Acinetobacter species have emerged to cause many nosocomial infections, mainly in Japan and in other countries. Among those were A. soli. One of those infections was a disease called bacteremia. Bacteremia is a blood infection that causes signs and symptoms such as fever, chills, nausea, headache, hypotension, and elevated white blood cell counts. The infection's pathogen, once able to reach the bloodstream, starts to invade the blood vessels via the lymphatic vessels to start an infection. Sometimes these infections can be caused by other fomites, such as needles or through catheters. With the case of spinach isolations, spinach as well as other foods that may be contaminated with the bacteria can be reduced by properly storing the packaged foods at temperatures of about 4 degrees Celsius or at 15 degrees Celsius. However, data was collected linking that storing foods such as spinach at these temperatures, although reduced its abundance of Acinetobacter, it later introduced families of Enterobacteriaceae. However, it is also very important to continue packaging and storing commercial packaged foods at required temperatures to avoid food spoilage. This concludes the presentation. Thank you for watching.